It is a pleasure to introduce Professor Dan Feldman. Professor Dan Feldman is from the Department of Molecular and Cell Biology in UC Berkeley, and as part of our annual lecture, we speak today about the somatosensory cortex. Thank you, Dan. But before that, there is a ceremony. Tali, please go ahead and let the ceremony.
Well, I wanted you to say a little more, but so as you all know, we have a very special week that we have two very distinguished lecturers that had our lecturers that were also a married couple, which is the first time we do this. And it's a pleasure and honor for us to give the Helen lecture series in Computational Lecture Neuroscience Certificate of recognition and appreciation to Dan Feldman for his contribution to academic inquiry and exchange at the Interdisciplinary Center for Neurocomputation and the Edmond and Lily Safra Center for Brain Sciences in Jerusalem, Israel, the Hebrew University, signed by me as the director of the ICC and by Helen as the head of the PhD program. And we're really happy to have you here.
Thank you very much.
And please come again.
Okay, thank you, everybody. It is a real honor to visit. It's actually my first trip to Israel, and so it's been interesting both from a cultural point of view and also from a scientific point of view today. And I think, as Marla mentioned to you when she gave the talk on Tuesday, we feel a little bit funny as a couple to be invited to share a lecture series because we don't really work on the same projects, although we have a lot of overlapping interests in terms of circuit function, and I hope you'll see some of that today between my talk and hers. So my lab is interested in how cerebral cortex works. And of course, we've known for many years some of the basic backbones of the cortical circuit. This is a lovely image from Cajal from 111 years ago or more. And you would think that smart engineers looking at the circuit could figure out exactly what the cortical circuit does, but of course, it's much more complex than that. And there are many questions to be answered, I think, about how these circuits work and what they do for overall brain function. And in particular, we focus on primary sensory cortex. This is, regions of cortex that, again, you would think from the textbooks are understood. We know to a great degree what these do. The standard model is that these are involved in identifying particular features inherent in sensory stimuli, representing those features in the brain, integrating them over space and time, building up the basic feature representation for sensory inputs. But how circuits do this is very poorly known, in my view. And here is an example of some outstanding questions that we and many others hope to address with our work. How do cortical circuits quantitatively represent and process sensory information? What are the actual computations that are being performed in this network, and how are they being performed? And very importantly for us, these circuits don't emerge fully formed during development, but they emerge in a fairly immature fashion, and they are actively optimized and refined by sensory experience in the first few weeks or months or years of life. How does this optimization process work? How is information about the sensory world stored during this process or during learning? And we hope that in addressing these questions, not only will we understand more about what the cortex does, but we can begin to address diseases of cerebral cortex circuits that remain mysterious today, diseases like autism and epilepsy and schizophrenia, which fundamentally, I think, are processing disorders, circuit-level processing disorders. But we don't really understand at the mechanistic level or at the functional level what's wrong in these diseases. So that's our motivation. Okay. So we study all this in the rodent whisker system. Whiskers, as I'm sure many of you here know, are tactile organs that reach out and touch the world, somewhat like fingertips, and gather information about the location and identity of objects in the world. This is a nice video from Aida Hiso's group at the Weizmann Institute. Information gets from the whiskers to the cortex in a pretty well-defined pathway. There are 5 rows of whiskers on the face that we call A, B, C, D, and E. Each one contains 5 to 8 whiskers within it. And if you look down at somatosensory cortex in the body map, there's a whisker area. It's blown up here and here. you can stain the cortex for a number of different markers which show you clusters of axons or cell bodies, and what you find is that within layer 4, which is the input layer to cortex, cell bodies are actually clustered into these large groups called barrels, which contain about 1,000 neurons each, and there's one barrel that marks the center of a cortical column, one column for each whisker. So here, for example, is a cluster of cells representing the second whisker in the D row here, and the third whisker, etc. And there's this nice map of the A, B, C, D, and E rows of whiskers. And we know from many groups over the years, the basic circuitry here, there are clusters of neurons similar to this in the thalamus that represent the feed-forward input from the whiskers, for example, whiskers D1, D2, and D3. These contain excitatory neurons. Each neuron in a given cluster, like the D2 cluster, projects up to the corresponding group here, which is called a barrel, as I said, in layer 4 of S1. And then excitatory cells in the barrel project up to layer 2, 3, also down to layer 5, not shown here. And then these intracolumnar projections from thalamus to 4 and from 4 to 2, 3 turn out to be quite strong, but they're functionally important, although slightly weaker connections across columns in layer 2, 3 and down in layer 5 that integrate information across the whiskers. So that's the very, very basic excitatory backbone by which information moves. And because of this, if you wiggle a given whisker, like the D1 whisker, and you look down in cortex, and here I'm plotting schematically in color, where do cortical neurons spike within that somatosensory map? When you wiggle the D1 whisker, cells spike in the D1 column and actually just around it. If you wiggle the D2 whisker, cells spike in the D2 column and just around it, etc. So there's a crystalline, although somewhat overlapping, map of the whiskers that's a physiological map layered on top of this anatomical map. So that's the basic organization. So we know now from many groups a lot of details of what the circuits are within this area, and this is actually the reason that we work in S1. So this is an example of a highly selected subset of neurons where we actually know cell types and connectivity and synapse properties from one neuron to the next. So in the projection from thalamus to layer 4, not only are there projections onto excitatory neurons, there are also projections onto specific classes of inhibitory neurons, which make inhibitory synapses, in many cases with known functions, onto those excitatory cells. These cells also receive input locally. Likewise, in the projection from layer 4 to layer 2, 3, there's inputs not only onto excitatory cells, but onto subclasses of inhibitory neurons, making still other very specific inhibitory circuits, etc. So we know a lot about how this circuit works, and in a given column, we know there's about 10,000 cells. Okay. So the question is, what does this cortical circuit do? And people ask this question at many different levels. I'm going to focus in on one particular level here, and that has to do with not a specific question about how individual features in the world are represented, but with the generic observation, really I think a contention at this point, a hypothesis, that sensory information is represented in these circuits with very few spikes. And the term for this is sparse coding, which I'm sure many of you know. So we're going to focus on sparse coding, in which optimal stimuli for this column are encoded with very few spikes. Now, evidence for sparse coding in somatosensory cortex is quite strong, but it primarily comes from work in anesthetized animals, applying what the experimenter believes to be an optimal stimulus and counting the number of spikes that are produced. In a subset of cases, people have looked in awake animals that are sitting there passively, and they apply stimuli that, again, are hypothesized to be optimal stimuli in order to see how many spikes they produce. But there's been a general lack in the field, I think, of evidence until very recently from animals that are actually performing sensory tasks, seeking out optimal stimuli on their own, even if the experimenter doesn't know them, to ask how many spikes are really produced in these natural circumstances. So we're going to start with awake-behaving animals. How sparse is the coding really? And then I'll move on to what are the circuit elements that help make that coding sparse, and you'll see some evidence that I think it is actually quite sparse. Okay, so we asked the animals to find their own optimal stimuli, and we did this here by training animals to do a texture discrimination task. This animal reaches across a gap, moves its whiskers across a rough sandpaper and a smooth sandpaper. Its job is to determine the rougher sandpaper, jump on that side, and get a reward. And the animals do this quite well. And in fact, we can measure the psychophysical performance of the animals, and it turns out that they are very good at this task, qualitatively similar to human fingertips. Okay. So how do they do this? We want to use this behavioral paradigm to try to determine what is the fundamental elementary feature of whisker input that might be coded and processed by that cortical circuit. Is it the overall texture? Is it the whole movement? Is it something microscopic? Okay, so the first experiment we did was to train animals to whisk back and forth on these sandpaper surfaces, and we used a specialized laser-based imaging system to get a one-dimensional movie of the whiskers moving across the surface at very, very high spatial and temporal resolution. And what we found were traces like this. This is a position trace, position here, forward and back, of a whisker moving across time during one cycle of whisking as the animal pulls the whisker back and then moves it forward across the surface. And what you can see is that here during the forward motion, there isn't a smooth motion like in the sinusoid that an animal would have in air, but instead the whisker rapidly moves to some position on the sandpaper where it gets stuck for a little while, and then it moves to a new position and gets stuck there, and then slips to a new position and gets stuck there. And so we call these slip and stick events. And when you look at whiskers moving on surfaces, these are a very, very prominent component of whisker motion on surfaces. These events are quite discrete in time. Here's a series of forward slips identified during forward whisker movement of animals on many surfaces. And there's a discrete high-velocity component where the whisker slips to a new position. The same thing occurs in backward movement, etc. And in fact, these whisker slips turn out to be a very good cue for certain aspects of tactile features. So for example, if an animal whisks back and forth in air, and then the same animal on the next trial on a smooth sandpaper, on the next trial on a rough sandpaper, here in these acceleration traces on the bottom, I'm just using acceleration to identify when a slip occurs, and I've put boxes around the slips here, and you can see that there are more fast whisker slips on rough sandpapers than on smooth sandpapers than in air. I won't show you the statistics, but this turns out to be very robustly true. And so these slips are actually a strong candidate for how animals might distinguish different textures of surfaces. And we have studies on this, but I'm going to focus here on just the slips as an ethologically important component of natural whisker input that the cortex might be specialized to represent and process.
So this is opposed to stick?
Excuse me?
Is it opposed to stick?
Oh, no, I'm sorry. So I'm going to use slip as a shorthand for slip-stick combinations. They always go together. The whisker slips to a position and then stays there.
So this will also be true, the statistics, if you compare air, the sticks part.
Yes, so it's true that sticks also vary with roughness. Sticks are a little bit more complicated to analyze for various technical reasons. You know, for example, at the peak here, it's hard to identify sticks and slips, but they seem to be similar. Okay. One key aspect I should mention of these slips you can appreciate here. So this is a few cycles of whisking. It takes a couple hundred milliseconds. Animals, when they're doing the texture discrimination task, they whisk for a few cycles over a couple hundred milliseconds to gather the information they need. During this time, there is a fairly dense temporal series of slip events, bop, 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 bop. And in fact, on average, slips have a mean interval of 60 milliseconds. And that means that the natural stimulus as animals palpate on surfaces is actually a temporally dense series of discrete events. It's not a continuous event.
So are the slips just a function of the environment, or is the animal also whisking differently in response to the environment?
We think it's primarily the former. So the whiskers interact frictionally with the surface, and as the animal tries to move its whisker forward, it gets stuck frictionally. It has to, push a certain amount, and then the whisker slips forward, resulting in a high-velocity slip event. There is some evidence that when the whiskers get stuck, the animals might also push harder to get beyond that, so there's a little bit of the second component as well. Okay. So are these slip events actually encoded in cortex? And this was an experiment we did a number of years ago to show that they are. The experiment is to implant rats with an array of tetrodes to record single unit spiking in S1 as animals do this task. And then we're going to correlate the spiking of single units in S1 with the slipstick behavior of the whisker on the surface. Okay? This is a raster showing spikes of one single neuron recorded in S1, and we're recording here in layer 4 and layer 5, which are the thalamic input layers. There are 200 trials of the animals starting to whisk on the surface. Each black dot is a spike. I've aligned all the rasters on the red line, which is the first big slip event during that trial. Okay? And the bottom is the sum. So what you can see is that there's background firing all the time. Slips do, on average, evoke an increase in firing rate, right? However, on individual trials, there are much more often no responses than responses. So here, for example, is a whole string of trials where there were no evoked responses to the slip. On other cases like here, you can see a response. And when you look down and count the spikes, on average, the probability of a big fast slip, an optimal slip, driving a spike is only 0.1 over spontaneous firing rate. And in addition, when the responses occur, they're only single spikes. They're never bursts. And that means that for a single neuron, there would really be no way to determine whether a slip occurred on the basis of spikes, because this slip-evoked spike here is identical to this spontaneous spike here. So single neurons wouldn't really seem capable of detecting when a slip occurred in the environment. However, notice that there's a feature of this response, that this is a 20 millisecond time scale here. The period of evoked spikes is actually quite brief. So after a slip event, there's about 15 milliseconds or 20 milliseconds of elevated firing rate, and then the probability of firing goes back down. And this turns out to be true across all the neurons in S1, and about 75% of neurons in these input layers in S1 show these types of responses. So there's a sparse spiking on the individual neuron level, but across many neurons with a tight timing of all those spikes. Okay. We wanted to ask whether on the population level this meant there could be an efficient readout of slip information even though there was a weak representation in single cells. Okay, so here's an analysis we did. We had gathered the physiology data that I told you about with tetrodes recording groups of 3 or 4 neurons simultaneously at a bunch of different recording sites over different animals asynchronously. But we did an analysis of firing probability across jointly recorded neurons and found that the probability of joint spiking between 2 neurons was the same as the product of the individual firing probabilities, suggesting that to a first approximation, cells were behaving independently. And therefore, we made an assumption that if cells are firing independently, we can make a prediction about how this information about slips is represented across the population. Okay. So we had 90 neurons that we recorded those slip-evoked responses over 3 different animals. Here we're going to assume that those 90 neurons are all there in a single trial, and we're going to ask what's the ability of a network to detect whether a slip occurred on the basis of spikes in those 90 neurons at the same time. So we have 90 neurons. For each neuron, we have the measured slip-evoked PSTH. We simulate a trial by saying for each neuron, we draw whether or not a spike occurred based on the firing probability in the pre-slip period. And then we have a post-slip period where we, again, remeasure the firing probability drawn from the real PSTHs of each neuron. And we can take the simplest possible population measure we could think of, which is the total sum number of spikes. So we're modeling here a 20 millisecond window. What's the number of spikes in a window before a slip event and after a slip event? And using ROC analysis, we can ask whether there's a detectable difference in the number of spikes here versus here. And as you do that, of course, you can vary the size of the population by undersampling or oversampling your measured distribution. And what we found was that a population of 100 neurons was sufficient to give 97% accuracy in detecting whether a slip occurred. So even though an individual neuron was not very good, not good at all at determining whether a slip occurred, 100 neurons was actually very good. Now, why was this? This works because the slips are synchronous, right? The number of spikes in a brief window is a measure of raw firing correlation in the neurons. And so what this model is really doing is saying that the raw firing correlation in brief windows predicts slips accurately. And in fact, in the model, we could rerun the model with different time windows from brief to longer. And no matter the number of cells in the network, we always got optimal detection in a 20 millisecond time window. No surprise, that's the window that slips evoke spikes. Okay. So all this is saying that slips can be encoded by raw firing correlations or transient firing synchrony on this 20 millisecond time scale. Now here's an illustration of how we think this model may work, and this is based on data from 4 neurons that were actually recorded simultaneously from a single tetrode. Okay. So here are 4 neurons. The animal came in and whisked back and forth on a surface, and during that time we identified 5 slip events, bump, 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 from the whisker motion. Okay. And these are the spiking of the neurons. So the first slip event was associated with spikes from neurons 1 and 2, but not from 3 and 4. Right? And on average, the firing probability to the slip would have been 10%. And then between the slips, there's additional spontaneous or non-slip evoked spikes. The second slip event evoked firing of these 3 neurons within the brief window. This slip event, only this 1 neuron, but presumably neurons we're not recording from. This event, 2 neurons, etc. So what we have is, for each slip event, a shifting population of neurons that are firing together in brief time windows. And that's what gives the detectability for the spikes in the model that we've given. Does that make sense? So the idea is that you can recover information from this transient firing synchrony, even though the firing in each individual neuron is very low probability. You can do it because the spikes are precisely timed. Okay? And I think this is actually a representation of sort of a common way that people think of population codes in these type of networks. Okay, now we want to move beyond this and say, how could this code really work in a more detailed way, and how could the circuit build in the ability to produce this? And so we shifted gears to layer 2-3 of the cortex, where, as I'll show you in a bit, the code is actually also sparse, and in many measures even sparser. And here we wanted to get a measure not just of a few neurons recorded together, but of as many neurons densely as we could. So we imaged, using two-photon imaging, calcium signals from populations of neurons in a single column. So I think we might need the lights down here. But what you're going to see here is a field of neurons. Thank you. I think that'll be good enough. So we'll see a field of neurons in layer 2-3 of a single column. They've been loaded with Oregon green BAPTA dye. Oh, this is very difficult to see. I'm sorry. But up in the corner, the word stimulus is flashing. We're wiggling the whiskers. This is in a mouse, an anesthetized mouse. We're applying a slip-like stimulus to the whiskers. And the bright white flashes are calcium transients. And what you'll see, I hope, is that some neurons, like this one and this one, fire to every maybe fourth or fifth stimulus that's applied. And many, many neurons fire to only every 10th or every 20th stimulus. Okay? So even though we're applying a very high-velocity slip-like stimulus, you get only a small population of the layer 2-3 cells that are active. And this has also been seen in work from several other groups. This is a histogram of the firing probability measured in experiments like this across many different neurons, showing that most neurons actually fire very little at low probability to these large stimuli. And there's a tail of neurons that respond with relatively high probability. Now, one can look at this and say, because we know that neurons in primary cortex are tuned for stimuli, there's always the problem that if we didn't apply the correct optimal stimulus for a neuron, that, of course, we wouldn't drive that neuron well. And so the cells that we see in our sparse code that are not active are simply cells that we didn't produce the right stimulus for. Right? If we had found the right stimulus, they would be very active. And this is a major sort of experimental design problem, and how to get around this for these types of experiments. Yeah?
Can you explain again how in anesthetized animal you mimic sleep-like stimuli?
Oh, all we do is we apply a whisker stimulus that has the duration and velocity of a natural whisker slip that we measure under awake behavior to the whisker. The animals do not whisk themselves under anesthesia.
And you do just one, like separated in time?
So in this case, we give a single slip event separated, I think, 5 seconds later from the next one and the next one and the next one. Those are the multiple repetitions that you're seeing in the movie. This is in contrast to a lot of these calcium imaging studies that use very dense trains of stimuli in order to try and really drive strong responses. Okay, so there are some cells that are more active and some cells that are less active. Now, in these same movies, we gather spontaneous firing information. We're not wiggling the whisker at all. And it turns out that if you analyze the relationship between the probability that a cell responds to wiggling its whisker versus its spontaneous firing rate, there's a relationship where cells with the lowest spontaneous firing rates have the lowest response probabilities, and cells with higher rates have higher response probabilities. This is compiled over multiple animals. It's not just an artifact that some animals are more responsive than others, because if you analyze within a single field, you assign each cell a rank of the most responsive to least responsive to the whisker in its field, and a rank for spontaneous rate, you also get a beautiful relationship, right? So there are cells that are spontaneously less active, and those cells are the cells that are the least reactive to sensory stimuli, and likewise the most spontaneously active, the most responsive. And to me, this argues that we haven't just missed the optimal stimulus. There are subclasses of neurons in layer 2-3 of the cortex which are somehow, either via intrinsic properties or via how they are wired into the network, predisposed to be more or less active within this sparse code. Okay. And so in a very conceptual way, our idea for the sparse code can be summarized like this. That when you stand back and look at population activity across a large map, imagine 9 whisker columns in S1 here. If you wiggle a single whisker, it really is cells within the corresponding column that are mostly active. So much of the sparseness within this map comes from the same kind of mechanisms that you would imagine in visual cortex. Cells are tuned for a particular stimulus. In this case, they're tuned for which whisker is wiggled, and so neurons in that column are active. But if you look up close at that one single column, which is schematized here, then you realize that actually cells are very sparsely active in that column. They're not reliably active with high firing rates. And in fact, most cells, like the ones in blue, have a very low response probability, and only a subset of cells in green and red have a higher response probability. Now, what other stimuli could be represented in this little mosaic within the column? The best hypothesis that's out there, or the hypothesis with the most data, is that neurons differ in a column in detecting which direction the whisker moved. Did it deflect up or back or forward or bottom? And there is tuning for this. But in S1, it's incredibly weak tuning. This is very different from V1 direction tuning. So that maybe there's a 30% modulation if you were to plot an imaginary tuning curve here for cells tuned to one direction or another direction. There's about a 30% modulation with direction. So S1 is built, I think, differently than V1. Cells are tuned for a given whisker, but they're not very tightly tuned for a particular feature, at least not one that's been discovered yet. Instead, cells achieve sparse firing not by tight tuning, but by a low probability response rate. And that's our working model for how S1 is encoding this type of activity. Any character in terms of response probability? Right. As far as we can tell, yes, but we're a little bit limited on timing. So we've done this two ways. One is under anesthesia. We can leave longer gaps between the stimuli, and you still get the same ranking of response probability. The other way we've done it is in vivo, during the animal's naturally whisking. And in that case, we can ask, what's the probability of responsiveness as a function of the delay or the interval before that slip event? And there is a relationship, but it's very, very weak. But of course, you're limited there because there aren't long periods without slips. So as far as we know, we think that it's relatively invariant to that.
Dan, I just want to make sure that I understood. This animal, okay, it responds to a particular whisker, but doesn't it respond to different parameters? Let's say the speed of the whisker, the location of the touch, direct. So there are many parameters within this whisker. It's the same group of neurons that responds to all these sub-parameters. So how does it know whether it's moving to the left or to the right, or whether it's touched up and down?
Sure, absolutely.
What is the extra?
So I'm not showing this here from data we've collected, but across other labs, it's certainly been shown that neurons are tuned to some other parameters, but the tuning is relatively weak. So in this case, for direction, as I said, there's about a 30% tuning depth modulation, right? For the factors that you bring up, like tuning for whether the animal's whisking versus receives a passive touch, or where the whisker was in the cycle, all that tuning, unfortunately, has been documented down in layer 4 and layer 5, and it seems to be more of a layer-specific or septum-specific kind of tuning. Up in layer 2-3, we don't have that information yet, and so we don't know, I think. And this is exactly why we were motivated to do the spontaneous rate correlation, to ask whether there was something built into a subclass of neurons, a subnetwork of neurons, that was making them more or less responsive, independent of those kinds of features. So I'm actually not denying at all that tuning exists. I'm just saying that on top of tuning, there's another contributing factor to sparseness, and that's something about more active and less active neurons.
So what are the other cells that are silent most of the time? Why are they there?
I think that's a great question. I mean, I'm hoping that we don't really use only 10% of our brains, right?
That's what my teacher told me.
Well, I mean, we can talk about this more at the end, but some of the ideas that we think about, it's certainly possible that there are higher order integrative features of more complex stimuli that would drive some of these cells. That's possible. Another possibility is that they may represent some reserve pool for learning that might come online during plasticity or learning, and we're trying to look at that now. Okay, so one thing to note before I go on. So I'm calling these sparse responses because in response to a single discrete slip event, there's a low probability, 10%, of actually evoking a spike on an individual neuron within that 20 millisecond window. But because natural whisking is a series of slip events, if you think about the set of slips that occur over time as an animal palpates on a texture, even though there's only a 10% firing probability for each, these add up to an increase in firing rate during palpation on texture compared to not palpating on texture, right? And that increase in firing rate we've shown in behaving animals to be almost twofold. Okay, so one can call this sparse or not, depending on whether your focus is on the 20 millisecond time scale, in which case it's sparse, or on a 200 millisecond time scale, in which case it's less sparse. Okay, but the question is, how is the cortical circuit set up to be able to process information that contains relatively few spikes at these 20 millisecond time scales? And an experiment that really guided our thinking on this was done by the Sockman group. Actually, there were several experiments. These involved either awake or anesthetized animals in which they applied these slip-like events in the same way that we're doing. And they recorded from many, many neurons using cell-attached patching so that they could measure in the least biased way they could think of the spiking probability of neurons. And what they found, I'm just replotting results of their paper here, is that if you measure in the thalamus the response to one of these slip-like events, there's about a 30% probability of firing of cells in the thalamus. In the projection from thalamus to layer 4, this drops to about 15%. And in the projection from layer 4 to layer 2-3, this drops precipitously to 4 or 5%. And this argues that the reason that firing in cortex is sparse is not because of lousy encoding by the primary afferents, but because there are somehow active processes in the architecture here to reduce the number of spikes as you ascend the circuit. So what is it that causes that reduction, and how is this, could this code be achieved? Okay. So I mentioned that there's two aspects of the sparse code that have to work in order for this code to happen. One is you need few spikes. The other is that the spikes have to be precisely timed. And I just want to show you one slide on timing. We already know one circuit element that we know must be important in controlling the timing of spikes, and that's an element called feed-forward inhibition. Many of you know, I'm sure, that when excitatory cells make long-distance projections, there's a projection not only onto the excitatory targets, but very often the axon branches and innervates particular classes of inhibitory interneurons, often fast spiking or FS interneurons. And then these make inhibitory synapses onto the same cells. So this happens very strongly between the thalamus and layer 4, so that a thalamic spike will cause a direct EPSP in an excitatory cell, followed at a disynaptic delay by an IPSP from the FS cell. And in a collaboration with Massimo Scanziani's group, we showed that this was very powerful in controlling timing in S1. So in the slice, what we found was that if you recorded from layer 4 excitatory cells here, actually show you here, in current clamp and stimulate the thalamic inputs, there's a rapid EPSP, that's this synapse, followed and cut off by a very powerful IPSP via this pathway here. And this leaves only a few milliseconds for the cell to integrate all of its excitatory input and generate a spike if it's going to. Right after that, there are no spikes. And so the idea was that this feed-forward inhibition helps to reduce the integration time window of neurons and keep spiking precise. And we were able to show in vivo that actually it looks like this occurs if you simply take an anesthetized animal, record the spiking of one of these excitatory cells here with an extracellular electrode in layer 4. That's what I show up here. You can apply these slip-like stimuli over many, many trials. These are all the spikes that occur. This is the sum. And you can see you get a precise low jitter spike that's produced. That's what's blown up here and blown up in time in the control histogram. And that's a product of both the direct excitation and whatever shaping occurs by the indirect inhibition. If you then simply take that stimulus and apply it rapidly, like one stimulus every second or two, that adapts synapses. It turns out that synaptic adaptation in this pathway is much stronger than adaptation in this pathway. And so at the end of an adapting train, there's relatively less inhibition than excitation. And so you can ask, after an adapting train, what is the jitter and spike timing? And of course, there are fewer spikes because excitation is reduced. But you can see from the scaled fit here that the jitter of those spikes increases. So when you adapt out inhibition, you increase the jitter of these spikes, suggesting that feed-forward inhibition is actually helping to control and make precise spike timing. What is causing the reduction in the number of spikes? I will admit fully, and I'm a little bit embarrassed to say that we were partway through what we thought was an extremely exciting study on this when a beautiful paper came out from Carl Peterson's group showing exactly what we were finding. So I'm just going to show you Carl's data and not ours. This is an in vivo patch experiment where he takes, in his case, an awake animal that can either, he takes an awake animal, he patches from layer 2-3 pyramidal cells, and here are a bunch of different pyramidal cells that are shown. He then applies he has actually the animal whisking back and forth onto a pole, and whenever there's a contact event, he can measure the resulting change in the whisker-evoked PSP in those cells. And every cell that he recorded from here in these smooth traces shows a contact-evoked depolarization. But yet, very, very few cells showed contact-evoked spiking. That's what's shown in the bars, which are PSTHs. So this cell spiked, that cell spiked, a few cells spiked, a few spikes there. Most cells do not spike. Why is that? Well, the cells that don't spike still depolarize quite strongly, right? And this has been a mystery. You know, why all these cells are happy to spike? Why doesn't sensory input drive them to spike? Well, what Carl found, and what we found in our data also, was that if you look at the trial-to-trial variability in the individual responses, you find sometimes that the cell happened to be depolarized spontaneously before the contact event occurred. Sometimes it was hyperpolarized, sometimes very hyperpolarized. And contact events to hyperpolarized cells drove very strong depolarizing PSPs. Contact events to depolarized cells drove either no change or in some cases actually hyperpolarization. And if you plot for a single cell the relationship between the amplitude of the evoked response and the pre-contact spontaneous VM, there's this beautiful linear relationship that has an intercept. The intercept is the synaptic reversal potential for that response. And reversal potentials that are down close to -70 indicate reversal potentials where inhibition is dominating over excitation, right? And so the measurement that he made is summarized here at the bottom, that you can measure the whisker-evoked synaptic reversal potential for a variety of layer 2-3 cells shown in the open bars. And almost all of these cells have reversal potentials that are actually below spike threshold for those neurons. So the reason that few cells spike in S1, according to this model, is a question of EI balance. In cells that are wired up to have more inhibition than excitation, you get a little bit of depolarization, but never a spiking, right? But in cells in which EI balance is higher, there's more excitation, you may be able to reach spike threshold and actually get a spike. So this is what we now call the EI hypothesis for sparse spiking in S1. And the key here is that the variance that I told you about between more responsive neurons and less responsive neurons might map onto subsets of neurons that have high EI balance and low EI balance. So is this true? We wanted to test this, and we focused on a slice to test this. Now, first of all, there's actually very powerful feed-forward inhibition from layer 4 to layer 2-3. Simple experiment, S1 slice, stimulate in layer 4, record whole cell from pyramidal cells in layer 2-3. You get both direct excitation and indirect feed-forward inhibition. In a current clamp experiment, just like I showed you in layer 4, you end up with EPSP, IPSP sequences. If you use voltage clamp techniques to measure the amount of excitation and inhibition separately by measuring their conductances, then you can show that stimulation in layer 4 drives inhibition that is 7 or 8-fold greater than the excitation received by these cells. So inhibition is powerful. And it turns out to be powerful because there's a particular group of neurons here that are quite sensitive and produce very robust feed-forward inhibition. And these are the fast spiking cells again. And I'll just show you one experiment here where to determine this, we stimulate at different thresholds in layer 4. We record now cell attached from different groups of neurons in layer 2-3. We measure the probability that the stimulus evokes a spike in those neurons. And then we break into the neuron and characterize what type it was. And it turns out that fast spiking cells are activated to spike at the lowest stimulus intensities, much lower compared to pyramidal cells or to other classes of interneurons. So these seem to be an interneuron class that helps to generate powerful feed-forward inhibition.
Now, if you have a ratio of 1 to 4 between excitation to inhibition conductors.
I'm sorry? One to
This is the ratio between inhibitory and excitatory conductors. Would you not expect the reversal ratio to be much more negative than the one you showed us before?
These are experiments that are done in slices where there's no spontaneous activity, and we're just looking at the isolated layer 4 to layer 2-3 circuit. In vivo, with the whisker input, of course, you activate that circuit, but also recurrent circuits and lots of other things. And so I'm not looking really for an exact quantitative match. I'm just trying to, you'll see that we're going to try and look at the strength of feed-forward inhibition to see whether it correlates with more active or less active cells.
Can you say what is the delay between inhibition and excitation to the L2-3 steps?
In vivo or in vitro? Either. So in vitro, it's cleanest because the noise is lowest, and you get about a 2 to 4 millisecond delay, I would say, between the two. In vivo, the situation is a little bit more complicated because it varies with the stimulus that you apply, and there's actually a beautiful paper by Diego Contreras to show that depending on the direction of whisker movement, if you move the whisker in the direction the cell's tuned for, E leads I by a few milliseconds. If you move it in the opposite direction, they tend to be overlapping, arguing that the timing contributes to the response. Well, that's because in this case, this is somewhat of a special case. There's a population of inhibitory neurons in layer 4 that actually make monosynaptic connections onto layer 2-3 as well, and that's what contributes to this little split here. Okay, so here's what some of these fast spiking cells look like. Now the question is, can we test whether this EI hypothesis for sparse coding is true? Is it true that the more active cells receive a higher EI ratio? And so the experiment we do is to go to the S1 slice. We're going to stimulate in layer 4 electrically like I showed you, and we're going to record responses in layer 2-3, but this time we're going to record population responses using 2-photon calcium imaging again. Okay? So we load layer 2-3 with argon green BAPTA1, I should put down here, OGB1 dye, and this is an example of layer 2-3 that's been loaded with a loading pipette here. And then we zoom in on a little box, and that's what's shown up here. And I'm sorry, the cell outline's a little hard to see, but these colored boxes are the regions of interest that are centered on different cell bodies. Okay? And the first thing we wanted to do was to make sure that we could detect single spikes of neurons, because we want to measure the ability of layer 4 to drive spiking to recruit these cells. And so this is an example for one neuron of the calcium signal measured in that neuron in response to layer 4 stimulation at these different time points. Versus whether or not the cell spiked, measured by a cell-attached electrode on exactly that same neuron. And you can see in this case there's a perfect correspondence between the appearance of a spike and a nice calcium signal to show it. And on average, we could show that we get about 85% detectability of single spikes with this technique. Okay, so that's the resolution. So how are we going to measure this? What we're going to do is stimulate in layer 4 at gradually increasing intensities. The lowest intensities, like the intensities that are, in this case, double the intensity that just produces the tiniest measurable field potential response, produces no measurable spiking in any of the neurons that we're monitoring in the field. But of course, as you turn up the stimulus intensity here to 4 times threshold, a few of the neurons start to spike, but others do not. If you turn it up even more, now most neurons are spiking, but a few are not. Okay? And if you look at how individual neurons are recruited, they look like this. Okay, so I'm plotting for a single neuron here the probability of observing a spike to our layer 4 stimulus versus the intensity that we're stimulating in layer 4, normalized in a way I can talk to you about if you want. But basically, individual neurons will never spike below some threshold, and then there's a threshold where they sharply begin to spike, and then very soon they're firing at some maximal amount. That is, single cells have sharp thresholds for recruitment. But as has been seen in a few other systems, across different cells, there's a range of recruitment thresholds. So here the gray lines show you cells that were all measured in one field. Some of them had low thresholds. Some of them had high thresholds. The black line is the integrated thresholds across all the preps that we looked at. There's a range of thresholds across neurons, from the early spikers, the most sensitive spikers, to the least sensitive spikers. My hypothesis would be that from this one subprojection, this small part of the network, we're not studying the whole network, that these may be more active cells. They have the lowest thresholds to spike, and these are the least active cells because they have the highest thresholds. So, first of all, is it reasonable to think that there are cells that form some kind of intrinsic subpopulation of high and low response probability? Or could it be in vivo that because of where we happen to stick our recording electrode, we're activating the excitatory inputs onto some subset of cells? Those are our low threshold responders. And we're missing the inputs onto other cells. Those become our high threshold responders, right? That would be all cells are uniformly wired, but based on where our stimulus electrode is, we're seeing some more active than others. Yeah, it's about 100. It's about 150 microns, and it's within a it fills about half of the layer 4 barrel. So we think we're activating, a chunk of maybe a quarter or half of the individual layer 4 barrel. But of course, we're playing with stimulus intensity, right? Absolutely. So let me show you. Right, exactly. So let me show you this experiment. Okay, so to get at this, we wanted to know whether the more active cells, for example, might be pre-wired to be more active and not just reflect something like the location of our electrode and the stimulus thresholds, exactly. So in this experiment, what we do is we have one stimulation electrode in the layer 4 barrel for the column that we're imaging. This is the projection I showed you in the last slide. But in addition, we put a second electrode in the neighboring column. Now, we know these cells project 85% of their axon to layer 2-3 of their own column, and then there are horizontal projections from one column to the next. And there are only about 10% of axons, actually 7%, that go out of that column into these cells here. So we think that we're activating a separate set of inputs, okay? And what we're going to do here is I'll show you here for, I guess this is 14 cells, all imaged in this one column. This is I've ordered them in terms of their order of recruitment on pathway 1, the home pathway. That's in black. So here's a cell that was activated at the very lowest threshold, another low threshold, low. Now we're getting to the middle thresholds, and these are high thresholds, and this cell didn't respond. Now I'm going to plot the response to exactly the same cells, but the stimulation of this completely separate set of inputs. And what we see is that all the thresholds are over to the right, but the order is largely preserved. This was the most sensitive cell. Actually, these 3 were the most sensitive cells. These are less sensitive on that pathway. These less sensitive, that actually was the most here. And then these are all systematically less sensitive, and again, that one didn't respond. And so if we compare the ranking of sensitivity of cells to those 2 different pathways, there's a correlation between the ranking on one pathway and the ranking on the other. So we don't think it's a matter of just where we put the electrode. Of course, that helps determine it. But in addition, again, we think that there are cells intrinsically wired in to be more active and less active. Now, what's making them more active and less active? So here we did the imaging experiment. We identified at a given stimulus threshold cells that were spiking and cells that were not. So we have we're calling them here spikers and non-spikers. Okay? The spikers are the low threshold, more active cells. And after we determine whether they're spiking by imaging, we do a whole cell patch, we break in, we use voltage clamp to measure excitation and inhibition to ask what the ratio is, what the EI ratio is. And so the results are shown here. Let me show you actually first this. If you measure EI ratio as a function of time after layer 4 stimulus, the spikers have a higher EI ratio than the non-spikers. Why do they have that? They have that because both spikers and non-spikers turn out on average to have the same amount of excitation that they receive, but they vary in the amount of inhibition. And the non-spiking cells receive more inhibition than the spiking cells. If we take the conductances measured in each neuron and we play them through a very simple single-compartment passive model to predict how much depolarization they would produce, these conductances predict a stronger depolarization for the spikers than for the non-spikers. So this would argue that the EI hypothesis might actually be reasonable, and that that there's variation across cells in the amount of inhibition that these cells receive, and this is one of the determinants that makes subclasses of cells more likely to spike and less likely to spike. Okay, that's what I'm summarizing here. Okay, I'm not going to give you the second part of what I was going to tell you about, which is plasticity, but let me summarize it for you because I don't want to make you guys wait. We just looked at the performance of what you might say is the normal mature circuit, right? But as I mentioned, these circuits don't form fully functional. They're strongly sculpted by activity. And like these other maps, the whisker map is very strongly sculpted by activity. Most of the research on how sensory statistics shape cortical circuits has really focused on excitatory circuits. For example, well, actually, let me show you one slide here on how these cortical circuits are plastic. You have a normal whisker map in which individual whiskers activate neighboring overlapping columns. But if you trim a subset of whiskers for just a few days, let the animal experience the world with this reduced array, and then remeasure the map, what you find is that the representation of whiskers that were trimmed weaken and shrink within layer 2-3 of S1, and those that were intact can strengthen and expand. Okay? So it's been observed by several groups, including ours, that there's a number of circuit mechanisms that mediate this. And because this plasticity takes place in layer 2-3, the focus has been in layer 2-3. So we know, for example, that in these deprived columns, the excitatory input from layer 4 to layer 2-3 pyramidal cells weakens, and it weakens via a process that looks like cannabinoid-dependent LTD. So LTD, long-term depression, might be a Hebbian mechanism for weakening responses that are not relevant to the animal. And likewise, other groups have found evidence that Hebbian mechanisms like long-term potentiation or synapse sprouting might be responsible for strengthening representations. But all of this is work on excitatory circuits. Given the importance of EI balance, we wanted to know, how does experience shape inhibition? Does it shape it in a way that somehow is matched to excitation? And here, I'm sorry that I'm running out of time for you, but let me just encapsulate this for you. You can use slice techniques to measure the strength of not only excitatory inputs, but also inhibitory inputs onto these cells with the classic feed-forward inhibition. You can make these measurements in columns that have had normal whisker experience or that were deprived of whisker experience. And you can measure the functional strength of excitation and inhibition. And the bottom line, it pains me to skip through good data, but here's the bottom line, is that when you record in a deprived column and a spared column, measuring layer 4 evoked excitation conductance into layer 2-3 pyramidal cells versus inhibition, you find that deprivation decreases direct layer 4 to layer 2-3 excitation. That's the cannabinoid-dependent LTD I told you about, we already knew about. But in addition, it strongly reduces net feed-forward inhibition. And it reduces this, and here I won't show you the data, because it very powerfully reduces the excitatory drive onto FS interneurons in layer 2-3. That's offset by an increase in the output of layer 2-3 neurons, but the net effect is a reduction in inhibition. And if you calculate on a cell-by-cell basis the ratio of excitation to total conductance, or excitation to inhibition, you find that in control columns, which are gray dots here, there's a broad range of EI balance across different cells, but you can calculate an average for the population. You can make those same measurements in deprived columns. Deprivation reduces E and I in parallel so that the mean EI balance across the population is maintained after deprivation. And so really, you have very powerfully plastic inhibitory circuits, but also powerfully plastic excitatory circuits, and somehow plasticity is yoked to maintain EI balance. And as a little teaser at the end here, let me just say, we can do this not just in the slice, but we can do it in vivo. With whole cell patching in vivo, same experiment now, recording whole cell from layer 2 through pyramidal cells in voltage clamp. Now we're extracting whisker-evoked excitatory and inhibitory conductances. The circuit is much more complicated than just 4 to 2-3 feed forward, right? But nonetheless, we see that after deprivation, deprivation produces a reduction in excitation and a corresponding reduction in inhibition. Okay? And again, the balance is preserved. So let me summarize what I didn't tell you by saying that inhibitory circuits are not only active players in shaping sparse codes and sensory computation, but in addition, they're active players in plasticity. And they're active in a particular way, which I find very interesting. During the first few weeks of development, as experience is shaping the circuits, experience is affecting excitation via what we typically think of as synapse-specific actions at particular excitatory synapses, like LTP and LTD. But it's also shaping inputs into inhibitory circuits in some way so that EI ratio is maintained. And it turns out the dynamics are very interesting. I didn't even plan on showing you this, that deprivation reduces inhibition more quickly than excitation, and then excitation catches up, so that during deprivation there's a period of imbalance, which is a compensatory phenomenon, and then this rebalancing that I showed you, so that that balance is maintained after plasticity. And we posit that one reason for that may be so that you can still get efficient computation of sensory information even within reorganized maps. Right? Otherwise, if you disrupt EI balance, you might not have appropriate processing. So I planned too much, but I'm happy that I shared with you what I was able to get through. And the people who did this work were a series of graduate students, David House, Shantanu Jadhav, Jason Wolf, and then in addition, some very talented postdocs, Lou Lee, Justin Elstrott, and Patrick Drew. Thank you very much.
Thank you, Dan. Also for discussion.
Just wanted to comment. There is a study by Satman showing that there are in layer 2-3 the geometry of the pyramidal neurons would vary between very small pyramidal cells to rather large, by a factor of 4 or 5 in terms of membrane area of dendrite. This on its own, assuming you will get the same input, would result with a more sensitive group of cells just because they have higher input resistance, smaller membrane area with the same number of synapses. So I'm saying that geometrical element rather than inhibition excitation balance could also give rise to a subpopulation that is more sensitive than others, just the geometry.
I totally agree. And in fact, I have to say that our guiding principle in looking at this is that there are going to be multiple factors that determine response probability. And if we can figure out what are the different factors that contribute, we'd love to do that. We're actually doing experiments right now to look at cell morphology. I can tell you that there is no correlation between input resistance and whether cells are, sensitive spikers or less sensitive spikers.
Moving animals naturally stimulate several whiskers at the same time, sometimes with conflicting or complementary or conflicting information. How does it fit in?
Mm-hmm. How does that affect this?
How does that fit in your model?
Yeah, we haven't dealt with that in the model. Actually, the behavior data that I showed you was from animals that were trimmed down to a single whisker corresponding to the column we recorded from, so that we could simplify the circuit. But I think you bring up an excellent point. There are very well documented suppressive interactions between whiskers, which are most obvious under anesthesia. But when the animal wakes up, there's much more complicated interactions. Some cells show facilitation. There's time-dependent phenomenon. There's some tuning that's been found for wave fronts of activity moving in different directions across the pad. So there's the potential for a lot of specific circuits that could do interesting things here.
I have a question about the behavioral experiment that you described at the beginning when the animals compared two sandpapers. So there's a difference in the distribution of slits. And the question is, and you've shown that animals can distinguish between these distributions, but my question, can animals distinguish between, or are they sensitive to the particular types of the slits or only the distribution?
Yeah, so we're doing those experiments right now. This is turning out to be trickier than I thought, but here's what we're trying to do. So one way of phrasing that same question would be, there is a very nice work from Matthew Diamond and others showing that as an animal whisks for a couple hundred milliseconds on a surface, the appearance of all these slips and other slip-like events on there leads to just a greater overall mean velocity or kinetic energy, right? And he hypothesizes that mean velocity might translate into just an overall mean firing rate. And so animals might simply use firing rate to distinguish this. We look at this and say that if you look at correlated firing in 20 millisecond windows, you actually get more sensitivity in being able to distinguish different sandpapers. So which might the animal be using? So what we're trying to do now is to express channel rhodopsin in these layer 2-3 pyramidal cells, which actually works great. And then to have animals do the texture discrimination task, rough versus smooth. And then use channel rhodopsin to add spikes to the spike trains as the animals are whisking on the smooth sandpaper. Now, we can add them in particular temporal structures, or we can add them in a less structured way, and we can ask, in which case is the animal more fooled to thinking that it's a rough surface? So I don't have an answer for you yet, but I think it's possible to get that answer.
At the same time, only very, very few neurons are fighting.
Right.
So where does all this increase come? And all these synapses have to be active somewhere.
I think the synapses are active. So the stimuli that he's using, or the contact events that are generated in his case, are also in the ballpark that they should make about 10% of the neurons spike. And I think the numbers in his papers are often between 5 and 10%. And that means that there's going to be more feed-forward synaptic activation of layer 2-3 than there is recurrent activation. So let's just make the simplifying assumption that we're looking largely at feed-forward activation. He finds that when you measure the reversal potential, for the synaptic reversal potential, the cells that have, this is totally unsurprising, right? The cells that have the reversal potential greater than spike threshold are the ones that spike, right? And so the model is that even for weak stimuli or non-optimal stimuli, or even for the cells that don't fire, they're receiving excitatory input. It's just that they're also receiving a lot of inhibitory input, and that depolarizes the membrane, but then clamps it at a reversal potential which is less than spike threshold. So that's the model. I think that's a reasonable model, actually. When you look at the spike thresholds of neurons, neuron spike threshold or intrinsic excitability doesn't seem to predict whether it will spike or not to the stimulus. So it doesn't seem to be intrinsic. It seems to be the cell's location in the network or the strength of the synapses it receives. I'll make an exception actually for this point about morphology, et cetera. And in that Sockman paper, there was an argument that the more synapses a cell has in general, maybe the greater sizes, the more likely it might be to spike.
Maybe enough question. Okay. So, happy Hanukkah to everybody.